Hi class, this is a review session for chapters 10, 11, 12, How Cells Divide, Meiosis, and Patterns of Inheritance. On the smaller scale, as far as cells go, the smallest types of cells are going to be prokaryotic cells. These are bacteria cells, and the way that they're able to replicate is through a process called binary fission. It's a relatively simple process where they are going to replicate their genetic material. It moves to the opposite sides of the cells, and then the cell divides. There's some limitations as far as genetic variation. It's very limited. So ways that they can get genetic variation is from a nearby bacteria cell or another dead bacteria cell, or even through just random mutations. But prokaryotic cells do not undergo meiosis, so that for us, we do undergo meiosis, so can plants. And so that's how we can get genetic variation there. But as far as prokaryotic cells, there are limitations, but there are ways to get around that. For eukaryotic cells, we do have what we call chromosomes. All that chromosomes are is just really compactly condensed DNA. And so that has to happen because DNA is really, really large. And so to be able to fit it inside of a cell so that we could then make a copy of a cell, the only way we could do that is if we were to shorten or condense our DNA. Another really good distinguishing vocabulary word I need you to remember is somatic cells and gametes. Gametes is not on the screen, but so for anything that we consider a body cell, like skin, tissues, we call those somatic cells, and then sex cells like sperm and egg, we call those gametes. You may have seen this before. This is called a human karyotype. This is how we are able to lay out chromosomes, and we can see if you have maybe more unfavorable genetic conditions. This is something that they can do, and they can look at and see if there's anything like a disease or something. They can kind of tell you what's going to happen. At the 23rd chromosome set, this is where we distinguish if it is male, female, but just understand that there can be other combinations. It's not just XX, XY. You can have XXY, XXXY, XXYY. There's a lot of, I guess, variations that we might not be as familiar with, but just know that it's not always just XX or XY. We already went over how chromosomes, I guess, are set up and why they're set up in that way. For RNA and DNA, you might be familiar, but just remember that RNA is single-stranded, DNA is double-stranded. As far as how chromosomes, or you may be more familiar with how chromosomes look on the right than you are on the left. So these are chromosomes. The one on the left, these are chromosomes, but then they are replicated, which now we have two. And the reason that you have two is because then the cell needs to divide and it needs to get its own little chromosome. But just know that these over here on the left-hand side, these are chromosomes. The way that they're kind of attached to each other is here in the center. They have the kinetochores, which is proteins that helps them stay connected. Also, if you have two chromosomes, we tend to refer them to being sister chromatids. In the cell cycle, think of this as a life cycle of the cell. And so the biggest part of the life cycle is this one, interphase. Notice how interphase is G1, S, and G2, which is all of these. Here's G1, S, and G2. So all of these three is interphase. So this is the longest part of the cell cycle. There are a lot of things that happen in each of these parts, and we'll go over those a little bit more in detail. The shortest parts are going to be over here, the M phase and telophase. M phase has all of these. I wish this blue part said cytokinesis. That's a little bit misleading, but it's right here where it says cytokinesis. So M phase and then cytokinesis will be at the very end. M phase, you may remember, there is an acronym that most people use, which is PMAT, prophase, metaphase, anaphase, telophase. So that might help you if you need to remember how or the order that these occur. This pro-metaphase is an additional phase that they have in your book, but if you do remember at least prophase, metaphase, anaphase, telophase, you should be okay. So within the cell cycle, please remember that this is for eukaryotic cells, so these are cells that do have a nucleus. The three parts that we mentioned were interphase, were G1, S, and G2. In S phase, this is where your DNA is replicated. This is a really important phase that I need you to remember. So S phase is where DNA is replicated, but of course the other phases are important too. G1 is growth, that the cell is growing, and that's also the longest phase. And in G2, your organelles replicate. Organelles are things like mitochondria, Golgi apparatus, lysosomes, so those are organelles, and this is when they get replicated. For the actual mitosis or M phase, this is the prophase, metaphase, anaphase, telophase, or prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis is when the cell actually will divide and have two identical daughter cells. That's a really important thing that I need you to remember. At the end of mitosis, you will have two identical body cells or somatic cells. As far as how long it takes, it does depend on the organism. Something that is much smaller will take a lot less time versus something that is bigger, or it also depends on the type of cell within the body. For example, the fruit fly embryo, it takes about 8 minutes for the cell cycle to complete. Whereas other ones, it can take, like mammalian cells can take up to 24 hours. So it really does depend on the organism. So we already went over this part of interphase, and we went over the kinetochores. No, we haven't. We haven't gone over the kinetochores. So when I show you a picture in a little bit, I'll go over what they look like. So we have our chromosomes, and then they're connected by a protein, but then we have what we call the kinetochore on the opposite side, and this is how these two chromosomes will eventually be pulled apart and how they go on the opposite side of the cell. I'll show you a series of pictures so you can be a little bit more familiar. For interphase, again, this is the cell growth. This is when the DNA is going to get replicated and it's going to get ready for division. In the center, we have the large nucleus, and the centrioles are starting to appear. During prophase, you'll have your chromosomes, they're condensed and they become actually visible. Like if you were to look at a microscope slide, you can see the actual chromosomes, which is actually really cool to look at. And also notice that you still have the nucleus visible, but that the nuclear membrane will start to dissolve. For metaphase, this might help you a little bit, but for metaphase, just try to remember that they met in the middle. So notice all of our chromosomes are aligned right at the middle, and then at the next part, anaphase, they start to get pulled apart going on the opposite side of the poles. When I was in high school, I was taught for anaphase that ana went away because they were getting pulled away from each other. And then finally you have telophase. This is slightly different from anaphase. Let's go back to anaphase so you can kind of see the difference. Anaphase, telophase, anaphase, telophase. Slightly different, but you can just see that the chromosomes are farther away from each other. For cytokinesis, it's different amongst cells because for an animal cell does not have a cell wall. So during cytokinesis, they form what's called a cleavage furrow, versus in a plant, they have the additional cell wall, and so they can't really pinch the cell the same way. So what they will do is they will form what we call a cell plate, and that's during the cytokinesis part of the cell cycle. Some controls that we have within the cell cycle, for some of these you can't stop them. Right here it says the cell cycle has two irreversible points: replication of genetic material and the separation of the sister chromatids. So after that point you cannot go back. It's going to complete the cycle. But there are, checkpoints, right? They have checkpoints, and at the checkpoints it can check. That's why we call them checkpoints. It can see if there's any mistakes, and if it is fixable, it will be fixed. If it is not fixable, then this process will stop and the cell will just be signaled to die. So the checkpoints, we have them here, the G1 to S. So right here, checkpoint, checkpoint. So this is where we have our checkpoints. And then this is what they check for. So for example, this is for cell decides to divide. And so if it doesn't meet the requirements to divide, it will stop there and the cell will be told that it needs to just end itself. Here it says the cells make a commitment to mitosis, and then also they assess the DNA if the DNA was copied correctly, is basically what that is saying. So just try to remember that this is the cell cycle is probably one of the more important things I need you to remember. Versus meiosis. Meiosis is going to be for sex cells or gametes. Unlike mitosis, where you have identical daughter cells, in meiosis you will not have identical daughter cells. Meiosis is all about giving genetic variation. That's why we look different from each other, because we have genetic variation. For diploid cells, we distinguish them by calling them 2N, and they have 2 sets of chromosomes. So 2N, 2 sets of chromosomes. Haploid cells, 1N or just N, and they have 1 set of chromosomes. So these are going to be examples like gametes, sperm and egg. Versus somatic cells, those are going to be diploid, and then we have a picture or a diagram for you on the bottom, and that's kind of how they look. So once you have a sperm and an egg fertilized, we form what's called a zygote. So a zygote is fertilized. So just try to remember that. That is on your exam. That zygotes is a fertilized egg. For humans, the cycle for sexual reproduction is that you have, and it has here, I'm glad that it has it here, the ends. So I have 2 haploid cells that fertilize to form a zygote. They have mitosis, I guess, because the child is growing, or actually, well, no, it's before the child is born, so it might just be that it's making all of the skin cells needed to make this baby. And then eventually, that's just keeps going in this cycle. Meiosis is similar to mitosis, but it also does have some differences, and one of the main differences is that it divides twice. I have meiosis 1, where it goes prophase, metaphase, anaphase, telophase, and then it will divide one more time in the meiosis 2. So that's why I have prophase 2, metaphase 2, anaphase 2, and telophase 2. So just remember, for meiosis, cell division occurs twice. And that's another way that you get more genetic variation, right? Here's one of the ways that you can get more genetic variation. You have what we call crossing over, and this happens in prophase 1. I'm not sure that I see that in the cell, or I'm sorry, in the slide. Crossing over, let's put that like that so that we remember. Excuse me. So crossing over occurs in prophase 1 of meiosis. This is where our chromosomes will touch each other at metaphase 1, and then at anaphase 1 they end up. So let's go ahead and distinguish that. Your metaphase 1. So right in between, crossing over. So we have, again, our chromosomes, they touch each other, and once they touch each other, they're able to exchange genetic material, and then it goes back. This does, I will tell you right now though, in the textbook it should say prophase 1. That crossing over does occur in prophase 1. And so this is just reiterating, this is one of the ways that you get genetic variation. Another way that you can get genetic variation is the random assortment or random orientation. We already know that this is metaphase because we can see that they're all lined up in the middle, but notice how they're all different. Like here I have blue, red, blue, red, blue, red, red, blue, red, blue, red, blue. So notice the way that they're aligned in the middle is different. So after metaphase, they get pulled apart. and then telophase and happens again. So, but just knowing that these are randomly lined up in the middle is another way that you get genetic variation. This is a great diagram that shows you the difference between meiosis and mitosis. So for meiosis, notice it says prophase 1, metaphase 1, and over here it doesn't. It just says prophase, metaphase, anaphase, telophase, and then that's it. So I have prophase 1, metaphase 1, anaphase, telophase, cell division. Look, we can see our crossing over starting to occur. And then meiosis 2. So notice again here, this is where it stops. I have 2 identical daughter cells. These are 2N, which is diploid, and they are identical again. And over here I have 4 haploid daughter cells. So very different. In some ways, I mean, they are similar, but they also have their differences. So the final result for meiosis is you're going to have 4 cells that are haploid. In animals, they turn into gametes, which again we said were sperm and egg. For plants, fungi, and other protists, they divide mitotically. They produce greater numbers of gametes, adults with varying numbers of chromosome sets. So this is probably the biggest thing that I need to remember. We have 4 daughter cells, they're haploid, and they're all different, versus in mitosis you have genetically identical cells. Some errors that can happen, you can have non-disjunction, the aneuploid gametes, and some of the most common things I think they said was abortion, that this could happen randomly. I'm not sure how. It says that it is very common, and I'm not like extremely familiar with how frequent this does occur, but yes. And I think by abortion they mean like a miscarriage. For non-disjunction, this is where your chromosomes, they do not move to the opposite side, and so this is a more popular one that can be an error. For this one, it says that gametes, they have missing or extra chromosomes. But just know that there are errors that can occur during meiosis. For the final chapter, patterns of inheritance, this is something that was observed early on, and we had finalized that it occurs within a species and that traits are transmitted directly from the parent to the offspring. And this was before the 20th century, right? That we had already kind of established this. The very popular scientist Gregor Mendel is the one that did experiments and kind of solidified some of the things that we know now. He did his research using pea plants, which is what you can see here on the right, and they do produce different flower and flower colors, and so that's how he was able to do his experiments. But just remember that he did use pea plants, and so the way that he would do his experiments is he would be the one that would pollinate them. So you might be familiar that plants have flowers. Well, not all plants, but some plants have flowers, and they do need to get pollinated. This is with a bee, with a hummingbird, bats, but he kind of went ahead and did the process himself, where he would get pollen and he would choose where the pollen goes on which flower. And so he was able to see the offspring that occurred. This is what we call a Punnett Square. And so it only crosses 2 variations of a single trait. So notice how they're all the same letters. Big B, little b, big B, little b. And I wish this picture said what the B's stood for. Well, I guess you can tell from this picture. So little b is for white, big B is for purple, and so they cross them. And also you need to remember that these 4 right here, this is saying that this is a female and a male, and this is going to be the probability for one of their offspring. So this is not saying that they had 4 offspring, this is the probability for one of their offspring. So if they have an offspring, they have a 25% chance of that offspring being white, and then they also have a 75% chance of the offspring being purple. We determine if it is white or purple based on the first letter. So notice this one is different. It's a heterozygous, this is heterozygous, this is homozygous dominant, but we go by the first letter, big B, so we know it's purple. Big B, purple. Big B, purple. Little b, white. So I think I go over these words in the next slide so you can kind of see. Yes, so homozygous dominant is you're going to have two dominant alleles. We call these alleles if you weren't familiar. The letters are alleles, which are traits. Big P, little p is heterozygous, so I have two different traits here. And then here, homozygous recessive, little p would be white. So be familiar with these words, homodominant, homorecessive, heterozygous, and what they mean. This is what we call a pedigree, or you might be familiar like a family tree. It shows you the lineages. So here I have 3 generations. I have my grandparents here, their children, and then their children. For this one, this is, I think, for a disease. Yes, this is for a disease, and so this is called dominant traits. So a dominant trait appears in every generation. When you read a pedigree, they will have usually a key, and so for males, in this one, they're distinguished by having squares, and for females, it is circles. If it's shaded, it means that they have the disease. If it's not shaded, then they do not have the disease. You can also have variation, like this one is recessive, meaning that you can have a carrier, right? So people that are carriers do not express albinism. So albinism is when you do not have pigment in your skin color, you lack melanin. And so these people that are carriers, they have melanin, meaning they have color in their skin, but they carry the trait. So if they were to have a child with someone that also is a carrier, the probability that they have a child with albinism is much higher. So just remember, carriers don't express that trait, but they do carry it. We also have what we call dihybrid crosses, where you can have multiple traits being crossed, but the same rule applies where when you are determining the phenotype. Phenotype means how the animal will physically look, or plant, whichever it may be. So phenotypically, like this one, we go by the first letter, the big R and the big Y. So this would be, what is this, round, and I'm not sure what big Y stands for. Well, I guess it just says it right here. Round and yellow. So just if you were to get one of these, you read it with the first letter. So for example, this one, you would say big R and little y. And then this is round and assuming green. I'm not actually quite, yeah, because this is wrinkled. So round and green would be like that. So, okay. Dominance is not always complete, meaning that there's not just dominant and recessive. You can have a blend. Really common example is for flowers. You can have red and white and they produce pink flowers. So this is what we call incomplete dominance. And then the last one is codominance. Heterozygotes show some aspects of phenotypes of both homozygotes. So phenotypes, again, is how the animal appears physically, or plant, or whatever it is. And then the other one that they give you is type AB blood. Some things that can also change how an organism looks, or even their genotype, is environmental influence. So they can be exposed to something that promotes maybe a mutation or something of that sort. For this example, they say this cat, let's see, the color of the cat, which is a Himalayan rabbits and Siamese cats. Their allele produces an enzyme that allows the pigment production only at temperatures below, this should be degrees, 30 degrees Celsius. So their coat, I guess, is just something that is distinguishable because of environmental influence. And that's it. So hopefully this was helpful, and I will see you guys next time.